Hello everyone, Mr. Dundon here, and today we're talking activation energy, if it wasn't obvious enough by that beautiful, cute little doggy going up the slides. We're looking at enzyme activation energy. Before we do that, let's just remind ourselves that enzymes are biological catalysts that speed up reactions that otherwise might not occur. In the body, we are very interested in this, especially in digestion. So we eat large molecules, big carbohydrates, proteins, fats, and then we need something to break them down into their small building blocks so we can use them elsewhere in our body and recreate proteins and things that we need. So we break down carbohydrates into monosaccharides, proteins down to amino acids, and fats into fatty acids. So we also use enzymes in synthesis reactions. So we, in DNA replication, we're building DNA molecules. We need enzymes for that. For RNA synthesis and protein synthesis, we're using enzymes in all of those processes to put molecules together and bond them together. We also use enzymes to regulate lots of metabolic pathways. So for example, we might want to break down a molecule into smaller parts. We don't just do that in one big hit. We bring it down into small, little, manageable sections. So we might go from A to B, B to C, C to D, D to E, for example. The enzyme that controls going from molecule A to molecule B is very specific, and it's going to be different to the enzyme that controls breaking down B to C. And so if you haven't seen my inhibitors video, check that out in one of these corners. There'll be a little link. Check that out, inhibitors. We can then manufacture inhibitors and then control our enzymes to then control those reactions. So if we want to stop in an intermediate point, we just put an inhibitor, we manufacture an inhibitor to block the enzyme, and then it can't go from, say, B to C, and we've stopped it there. Here's a quick little video as a reminder from one of the previous videos in the series as well. So enzymes, we have substrate, we have the enzyme active site. The enzyme takes that substrate into its active site, has this induced fit, this conformational change, and then we produce some products. Okay, so that brings us to today's lesson where we're looking at activation energy. So what's that all about? Well, basically a reaction needs a certain level of energy to even occur, okay? We term that activation energy. So this activation energy is the top energy where the transition will occur from reactants to products. And this is going to be different for every single reaction, but it's the actual minimum amount of energy required for that reaction to take place. In this diagram here, on the y-axis, I've got free energy. I'll discuss that in a sec. And then on the bottom, we've got time, measuring our reaction over time. Here, I've got this little circle here to indicate our reactants, and then we've got this sort of curve. So free energy, what's that all about? Well, provided that our system is above 0 Kelvin, so minus a couple of 100 degrees Celsius, these particles are going to have energy. So they're going to have heat energy, which in turn means kinetic energy, which in turn means movement. So that means that they can either collide into one another and react, or given enough energy, the vibrational energy of those atoms can break bonds and then separate molecules apart. So that activation energy is that transition state where two molecules will form together or one molecule will break apart. So in this case, we've got A becoming B and C. Now, if that particular reactant doesn't make it up to that activation energy, we'll never get a reaction to occur. And some systems, we can never provide enough energy for it to reach that transition state to become B and C. And that's particular in biological organisms because they've got really controlled body temperatures. So for example, humans, around 37 degrees, we don't fluctuate much from that. So that's our heat, our free energy system that we don't really alter. Therefore, if this energy level is above 37 degrees Celsius, then those reactions will never occur in the body. That's where enzymes come in. So enzymes will lower this activation energy. So enzymes will create a curve. Oops, that was horrible. enzymes will create a curve like this. So the activation energy is now here compared to where it was before. So now we've had our enzymes reduce the activation energy required for that reaction to take place. So now these reactions are more likely to occur in the body, and we can get the products from them. So this activation energy, if we were to measure it, is the position between this dotted line where it starts and then to this top point here. So this is without the enzyme, and then the activation energy required with an enzyme is from here to here. So you can see it's much smaller. So the difference, the reduction in activation energy, is this point here. So that's our reduction. So that's our reduction in activation energy caused by an enzyme. So I'm just going to put a couple of notes here to remind you of the different scenarios. So blue is without the enzyme, and red is with the enzyme. Okay, so the activation energy for the with enzyme is the distance between here, and without the enzyme, it's this distance here. So lucky this is a video, so otherwise this would get very messy if it was just a single diagram. But we've got our activation energy without the enzyme, the activation with the enzyme, and then in green here, the reduction in activation energy. This distance here is the net output of energy. So this is the energy that has come from breaking down those bonds and releasing that energy into the free environment, into that free state. So that's the energy released from that particular reaction. So this would be an exothermic reaction. So hopefully that gives us a really good understanding of activation energy. So it's the energy required for the transition state. And then with enzymes, it becomes much lower, and therefore reactions are more likely to occur. And then if we undergo the reaction, you'll notice that it's also a little bit faster, occurs a little bit quicker, but the net output energy, regardless of whether you're using the enzyme or not, is the same. Okay? And remember that the enzyme's not change in any way throughout that reaction. So how does that occur? How does that enzyme actually lower the activation energy? Well, it does it in a couple of ways. One, it orientates the substrates. So you can imagine if you've got two substrates that you're trying to break down, and they need to collide in a certain orientation where bonds can form. If that was just to occur randomly, what would the probability of them colliding in the correct orientation be? Pretty unlikely. But if we've got something that can sort of catch one substrate and have it in the correct orientation, and then catch another substrate and make them in the same orientation needed, then we're going to increase the likelihood of that particular reaction. And it also puts the substrates in a position where the energy required for those bonds to make are reduced. So it's like the optimum. So they reduce the activation energy by orientating substrates. The other thing that happens is that the enzymes sort of put pressure on those substrates so that those bonds can break, or bonds can break and new bonds can form. So there's a small bit of pressure on the bonds of substrates. So not only are we reducing the activation energy required for the reaction to occur, but we're also increasing the probability of that reaction occurring just by better orientating the substrates. So hopefully that explains everything about activation energy. Again, if you like the video, give it a thumbs up. Make sure you subscribe, because these videos are all related to biology, probably at a high school level, and so you can use these for all the upcoming topics. And yeah, catch us on all these other social platforms as well, on our website, and you can find more videos from myself.